All right. Good morning, everyone. Thank you so much for joining us for day 2 of the BINA 2024 Community Congress. I'll waste no time with us getting started. I'll let Mike Nelson come up to introduce our speaker and get the slides ready to go. Thanks for joining us.
It's my pleasure to introduce Dr. Virginie Uhlmann, leader of the BioVision Center at the University of Zurich, and also who leads a bioimage analysis group at EMBL-EBI in Cambridge. And I think many of us can agree that bioimage analysis is the best kind of analysis. She also recently posted on the ImageSC forum about her group's work on a new Python package called Splinebox, and I'd like to get on my soapbox temporarily on that note and mention that the ImageSC forum is a place for both open source and closed source software, but if you want to be a community partner, you do need to be fully open source, and that means no non-commercial compete licenses, which was discussed yesterday. So I believe you'll be covering a little bit more on that topic when you speak. And so here, welcome, Dr. Uhlmann.
Right. So yeah, hello everyone. Thank you very much for the kind introduction. Thank you for having me here in Madison. It's really a great pleasure to be here. I know that there is a lot of image analysis happening in Madison, and there are some people working here who, I'm looking up to as like heroes of image analysis. So I feel a little bit as an imposter giving a talk in image analysis here. I feel like I have more to learn from the people around, but hopefully this is going to be entertaining. So as was mentioned, I have a research group at EMBL-EBI since 2018 that has been working on developing methods for bioimage quantification. And I'm also, since the beginning of this year, leading something called the BioVision Center at the University of Zurich that is a platform specifically dedicated to bioimage analysis. Since I have 2 jobs, I thought I would also give 2 talks in one, which I'm not sure is a good idea, but I think can work. And I think can also be a good way to essentially show how the sort of method development side of image analysis feeds into the tool development side of image analysis that is equally important, and that needs to get, I think, a lot more attention than it is getting now, as Beth very well pointed out yesterday. So, oops, I have to click? How does that work? Somehow this is not moving to the, yeah, okay, that was, sorry.
Should just need
yeah. Oh yeah, sorry, good. So I know that I'm preaching to the converted here and that I don't need to tell you how important and great microscopy images are. But I thought I would keep this couple of slides I usually have in my introduction, because I think even for us who love bioimages, it is good to sometimes just pause and reflect on the fact that I would argue bioimages are the sort of primary type of data that biology acquires, right? And you may recognize some of this from like textbook on the history of microscopy. These are often kind of mentioned to be the first microscopy images ever generated. I mean, these are obviously not digital images, these are hand drawing, but this is really kind of the first type of data that people acquired when they were doing biology, right? Before we had omics, before we had genetics, before we had all of these things. And one of the really interesting things about microscopy images is that it was there at the very early days of experimental biology, but it also kind of evolved with the field, right? And these are maybe the sort of bioimages that we are more familiar with nowadays that have nothing to do with the ones I showed you before in terms of scale and in terms of amount of information that they contain. But I mean, these 2 things belong to the group bioimages, yeah? And I think this also kind of ties back to one of the things that is really at the core of bioimage analysis, is that bioimage analysis, comparatively to, again, classical bioinformatics or any sort of omics analysis, evolved a little bit more slowly. And I believe that one of the reasons for that is that, okay, first, bioimages evolved very quickly along with biology, but also that somehow, bioimages were there before biology was quantitative, right? And I think there is still a bit of a sort of culture shift that needs to happen to really have everyone in biology embrace the fact that these things are actually a source of quantitative information, just as any other readout that we have on living systems is. And this is why bioimage analysis is so important, and why we a couple of people assembled into a little community around this topic. So there is this quote from Gene Myers from a paper he wrote in 2012 that was in this special edition of Nature Method around bioimage informatics. It was called, I prefer bioimage analysis, but this is my personal preference. And I let you read it, but I think it's, you know, this is kind of nice because I think this is really how the field was 12 years ago. Now I think things have evolved a little bit and we have consolidated as a much bigger community that, you know, has their own kind of conferences where we meet, their own venues where we publish, their own ways of acquiring funding and so on and so forth. But we are still kind of, you know, developing and growing field that has to position itself midway between classical computer vision, that is much more about working on what we call natural images. So there's images of like pedestrian in the streets, cars, cats, dogs, and so on, and bioimages that are images of living system. And I just want to illustrate, you know, this two videos here. What you see on the left is like the sort of classical computer vision problem, which is tracking people. I mean, one of the many classical computer vision problems, right? Tracking people at the start of a marathon line. And you can obviously see the similarities that you would have with something like tracking cells during gastrulation on the right here. But you can also get a feel for the fact that even though there is something conceptually similar, the type of questions that we are asking when we deal with microscopy images and the very nature of the data are very, very, very different, right? And it means that essentially adapting tools that come from computer vision to use in the context of microscopy data analysis is far from trivial. I think this was mentioned yesterday by Eric, and I like that very much. The fact that, you know, in computer vision, a couple of terabytes is considered like a very, very big and comprehensive data set. In biology, this is basically one light sheet experiment. So, I mean, there are quite a lot of similarities, and this is why bioimage analysis really has kind of branched as its own field that lives with very close connection, but independently of computer vision. And so the first bit I want to tell you about is some of the stuff that we've been doing in my group around developing methods in this bioimage analysis space. And I spent quite a lot of time in my life with mathematicians, so I kind of like to think about this. I think about things in terms of like high-level concepts, and it helps me to like categorize stuff. So I like to think about bioimage analysis as like a field that, you know, has different types of subproblems depending on the input and output that we are dealing with, right? And if we see things through this lens, there's sort of three big families I can identify of bioimage analysis problems are what I would call image restoration. That is anything where we start from an image and kind of end up on an image. So denoising, the convolution, all of these things would fall into this. Anything that is from an image to a partition of the image or objects. So of course, segmentation that we all love or hate, depending on the day or the time of the day, would be part of that, object detection as well. And last but not least, quantification, which is, okay, what happens when we have segmented stuff, right? How do we turn the partition of the image that we consider to hold information into numbers that actually describe this information? And I've been, from the beginning of my career, really, really interested in this last step, which, you know, I would selfishly argue has not given as much attention as it should have in the early days of bioimage analysis, but that is now having its kind of golden age. Because obviously, when you acquire images in the context of a microscopy experiment, your end goal is not just to kind of tell which pixels are cells and which pixels aren't. It's to actually gather quantitative information on this image and to have numbers that describe what is it that you see, right? And thinking about what are the right numbers, what are the right metrics to quantify, to turn visual information into something quantitative is not, in my opinion, a trivial problem, and is something that I believe is really, really interesting, both conceptually, but also like in terms of method development. So we've been interested in this, and among the gazillions of things one can quantify from microscopy data, I've been always very excited about morphology. I didn't know a lot about biology before I moved to EMBL, so now I could make a whole speech about how morphology shape is linked to function and is therefore really important in the context of living systems. The reason why I was interested in this in the first place was not necessarily because of all of the sort of functional underpinning of morphology, but about the fact that, this is a concept that we humans are very, very good at capturing and understanding, but that is really, really hard to boil down to something quantitative. Here I've put you 3 pictures on this slide that have absolutely nothing to do with each other, that I've just kind of found and that were fortunately CCBI. But the common denominator between these 3 is that if I was to show you any of these and tell you, yeah, there is some interesting morphology here, I think we would agree, right? And yet we wouldn't quite be able to tell what it means, but we both see it, right? And interestingly, now if we're saying, okay, so let's sit down and try and write a definition of what morphology means, that would cover these 3 use cases, but also like all the possible microscopy images I can come up with where we would agree there is morphology information, that would be really, really challenging, right? So there is something as a concept, a high level concept of morphology that is present in visual data, but that somehow we really have a hard time boiling down into something precise, right? It's a mix of texture, it's a mix of shape. It has to do with like how things are arranged with respect to each other. It's some sort of complicated combination of all of that. And this is something we see at all scales and in all sorts of living systems, right? So I got really interested about just thinking about that from a kind of abstract point of view, right? How can we think about developing methods and strategies to quantify what we refer to as morphology in an as modality agnostic manner as possible? Because this really is a property of like visual data that is common across different types of microscope, different types of experiment, different types of experimental systems. And so the research in my groups has been revolving around this. We have done work in really kind of bioimage quantification per se, which is thinking about how do we extract useful representations of morphology from images. So how do we move away from this image domain to a domain that is more quantitative and where we can really measure this thing we refer to as morphology. We did a lot of stuff on shapes because I'm fascinated by shapes. I will not talk too much about this today, but like this could be another talk. Then we also went into thinking about, okay, now, we've moved away from pixel grids and turned things into numbers, but like, how do we mine collections of such measurements? And this could be yet another talk as well, but this is something that I also find really, really deep and fundamental about morphology, is that this is also a phenotype that, has a lot of invariance properties, right? If we went back to the images I had on the previous slide and we're now rotating them around, the morphology information would be, so to say, unaltered. But if we were to zoom in, there would be new morphology information. So, what does that mean? Yeah? And this is really interesting to think about from a theoretical perspective. And then, I mean, it turns out that I've never sat in front of a microscope, and if I did, I would be utterly unable to do anything useful. But I've been fortunate to be surrounded by people who are actually really good experimentalists and ask really interesting questions. So what we've also been doing is to essentially work in collaboration with colleagues from the life sciences to try and apply these different techniques, or indeed develop new ones to explore interesting questions about morphology in the context of development, organogenesis, morphogenesis, and so on and so forth. So what I thought I would do here is just tell you about one specific project in the sort of bioimage quantification realm. It's not the latest one, it's from last year. Some of you may know about it, but I think this is also a good opportunity to tell you about one thing that is quite hot at the moment in image quantification, that is self-supervised learning. And that hopefully if you get at least one thing from this talk, it's just some basic notion of that could be useful. So when it comes to turning image information into quantitative metrics, I like to think about it as like roughly 2 strategies. There is one strategy that would be relying on what I would call handcrafted features. This is something that we have, I guess, all done at some point, and that is extremely valid. I will come back to this later on. It's basically to say, okay, I've segmented my objects, and then what do? I don't know what morphology is. I can't define it quite clearly, but what I can is to like measure the hell out of this object, right? And calculate everything I can out of it, like everything about its shape, everything about its texture, everything about whatever I like, and just assemble all of these measurements into a very, very big number. That's a very big vector, sorry, that will be essentially a feature vector describing the morphology of the object as I see it in my image. Again, this is great. It works amazingly well. And it is, as a matter of fact, hard to beat, as we shall see. But another approach that we can also think about, and that is enabled by the latest developments that have happened in machine learning and artificial intelligence, is to say, okay, why should I be the person who define these numbers when actually I could maybe just let kind of the data speak for themselves and have a machine learning system learn what are the good descriptors of the visual information I have in this object through training, yeah? And that would give me another vector of numbers that would be descriptors of morphology that, would be harder to interpret, but that's maybe more informative because there is no risk that I kind of forgot to add something because this has been learned from the data directly. So I just want to tell you a little bit about how this can be done. And I apologize to all of you in the room who know already about self-supervised learning. I didn't want to assume that everyone would know about it. If you do, you have 2 slides to do your image. If you don't, hopefully you learn something new. The simplest way of learning a quantitative descriptor of an image, like from a data-driven perspective, is to rely on a specific type of machine learning paradigm called self-supervised learning. And that is part of a kind of bigger subworld of artificial intelligence called representation learning. So as the name says, the principle of self-supervised learning is that you don't need labels, right? You don't need kind of ground truth annotation to be able to train your system. And as the name says as well, for representation learning, the goal of the game is to learn a representation of your image that is different than your image, but that is really informative as well, yeah? Sorry. And so in its simplest form, the way that you can implement self-supervised representation learning is with a category of models that we call autoencoders, and that very roughly work like what you see on this slide. So what I would do is to throw into my model an image. This would go through a first part of the model that does a lot of complicated nonlinear operations on it and convolutions and so on and so forth, depending on the backbone I've chosen, but essentially compresses this image into a low-dimensional vector of number that is called bottleneck, latent representation, embedding, depending on who you ask and on the time of the day. And then in a second step, it would take this low-dimensional vector and essentially try to expand it back in order to recreate the image you had as an input. Sorry. So you can get a feel for the fact that you do not need labels to supervise this thing because, I mean, the only thing it's supposed to do is to basically spit out what it got in, right? So in order to train it, what I can do is simply just say, okay, look at the output, compare it with the input. If it's very different, it's bad. If it's very similar, it's good. Go towards a direction in which it gets better and better, so more and more similar. And obviously, when this thing is trained, I'm not so interested in the output because, well, I mean, I knew it from the start. What I'm really interested in is this low-dimensional representation that is in the middle. Because as a matter of fact, what this system is doing is essentially learning how to compress my image, my original image that is whatever 512 by 512, sorry, into a vector of a much less number, a much smaller amount of numbers that is sufficiently informative, that holds sufficient information to be able to reconstruct back the input at the end, right? And this thing is in the middle is my feature vector. This is basically the equivalent of the thing I can build with my handcrafted feature, except here it was, oh, thank you. Oh yeah, and my bottle appeared here. This is very kind. I didn't notice, sorry. Whoa, magic things happen. And so yeah, this thing is basically the equivalent of your handcrafted feature, except it was obtained in a data-driven manner. So now the image you see in the slide, several of you will have recognized it probably. This comes from a data set called MNIST that is used in computer vision for benchmarking. It's a data set of handwritten digits, right, with different types of handwritings from 0 to 9. And I just want to use this to illustrate why this sort of approach is interesting in practice. So if we take the whole MNIST data set, so all of these images of digits, we throw them into one such autoencoder model to essentially turn each and every one of these images into a feature vector that is representative of what was in the image, yeah? Then we have this new representation space that is not the image space, but in which we have each of our data points and we can essentially explore what's going on. That's what you see on the right. So obviously, if my vector is more than 2 numbers, I will have to do some sort of projection to visualize it, right? So this is what happened here. And now what I've done is to color each and every one of these data points that used to be an image before it got turned into a vector, according to the digit that was contained in the original image. And the first thing that you notice is that same colors group together. This sounds really trivial, but it's absolutely not, right? This system was at no point told that it's even looking at handwritten digit. It has no kind of prior notion of the fact that it has several images with the same sort of information in it. It retrieved this, it kind of understood this through the process of learning a good representation of what was present in the image. And you see more interesting things that I've highlighted with red arrows here. You have a blue cluster and an orange cluster here that correspond to essentially digits, images with digit 1 and images with digit 7. These things are next to each other, which makes sense, right? I mean, morphologically speaking, these two things look alike. But again, this is not something that we added as prior information. This is essentially something that emerged from the representation that the system has learned of the image content. So this has existed for some years and got quite a lot of attention a couple of years ago. And together with my bioimage analysis partner in crime at EMBL, Anna Kreshuk, we started thinking about, okay, this is really cool, you know, could we do something similar, but in the context of biology? And I mean, of course, you know, encoding and written digits is only so much interesting for life sciences. But what if what we were trying to get to is like getting a representation of cells or getting a representation of organs or getting a representation of tissues in this process? And so we started thinking about that. And essentially we had the idea, all we needed was a data set. But fortunately for us, some other colleagues at EMBL were very, very busy in the previous years generating a fantastic data set that you see here and that is described on this paper that is essentially a whole 3D EM volume of an entire platinerase sea worm. So I didn't even know that platinerase existed before working on this project, but it turns out that this is a sea worm that is really interesting and that some of my developmental biology colleagues are extremely excited about because it does all sorts of kind of very unique things. And one of them is that it has very, very, very stereotypical development to the point that if you were to take several adult platinerates that have the same age, they would probably have, I mean, they would very, very likely have the same number of cells at the same place, yeah? So this is super interesting to study morphogenesis, to study kind of the structure and the organization of the animal and so on and so forth. And so what was done in this previous work was to essentially prepare a sample of one such organism, entire organism, and image in all of its 3D EM glory at a resolution that, you know, I could only dream about before seeing this. So here you just see a couple of slices. Now this is another slice that is kind of hopefully gives you a better sense of the amount of detail that we have. It turns out that, you know, because my dreams sometimes come true, not only this data set existed, but also it is segmented because, you know, why not? So we actually also have labels for each and every cell in this animal. I mean, not labels, sorry, masks, but we don't know what these cells are, obviously. The segmentation is not perfect, far from it, but I mean, you know, I'm not asking for anything more at this point. So we got really, really excited about this because it essentially means that we have a data set in which we have something like 20,000 cells where our developmental biology colleagues know or have some expectations about what these cells would be, but really nobody has gone through the hassle of going through this and annotating everything. And so it feels like, you know, we could unroll something very similar to what I showed you with the MNIST data set, but this time instead of handwritten images of handwritten digits, we have actual cells coming from electron microscopy images for this entire animal. So this is what we did. And this was a fantastic project, again, together with Anna, my dear colleague, and Detlef, who is one of our EMBL colleague who is a developmental biologist and platinerist expert. So he's really the one who's leading all of the biology questions and development around this. The work was carried out by Valentina Zinchenko, who was Anna's PhD student and who really did all of the data analysis and kind of biological discovery that I'm not going to talk about because I am the least appropriate person to talk about this part, but that is all in the paper. The other lead in this project was Johannes Huger, my PhD student, who developed the deep learning framework that I'm going to describe in the couple of slides that follow. So what we did here is really pretty much exactly the same thing as I showed you with the MNIST dataset before, except of course, it's not just a 512 by 512 binary image now. We're dealing with 3D volumes of electron microscopy images that are really, really big. So yeah, we can't just like throw this into one model without making our computer blow. And we therefore had to be a little bit more smart about it. But also, we kind of wanted to have a little bit of interpretability here in that we wanted to be able to kind of go back to the feature that our system has learned and be able to tell, okay, what is the most discriminative for, you know, this group of that group of cells that we notice? Is it texture? Is it shape? Is it this and that? One thing that I can say that kind of really was interesting for me in this project, coming back to my introduction, saying that, you know, I kind of feel like there needs still to be quite a lot of culture shift toward making bioimaging quantitative, is that when we started working on that and asked, you know, Detlef and or developmental biology colleagues, okay, what did you expect in terms of what the shape of different cell types would be? They basically told us, like, we have no clue. We never looked at that in 3D, which was quite interesting, because it really is a byproduct of the fact that these data are gigantic and essentially too big for the human eye to explore, right? So what we did here is to essentially not have one autoencoder system like I showed you for MNIST, but 3 of them that encode different type of information we take from these images. The first one is shape. So this is really just geometry information, devoid of any texture. The second one is what we call texture context. So this is essentially a lower resolution version of the texture of the entire cell. And what this means is that you don't see all of the tiny details, but you do have context as to where texture is inside the object. And the last branch is fine texture patches. And this is essentially you keep the texture at the highest resolution, but you chop it into cubes such that it can essentially fit into GPU, right? And that means you lose this notion of localization, but you see all of the details of the texture. Each of these different branches, they create a feature vector that we then concatenate into a big feature vector. It's not extremely small. It's a couple of hundred numbers, but essentially this is still much, much smaller than the tons of voxels we had at the start, right? So this is quite a compact representation. And then what we can do with this is exactly the same sort of stuff I showed you with MNIST before. So we can say, okay, now we have this new representation of the cells as they appear in the EM volume. Can we just explore that and see if this brings us to new discovery? Before I get to this point, I just want to tell you one more thing about the shape part, because this goes back to my point about morphology and invariances and this being really, really interesting. Shape is a really, really interesting phenotype as part of the different things that compose morphology, because this is something that you can't, you don't really want to treat as, okay, I want my autoencoder system to, you know how to encode shape and then reconstruct it perfectly. If it reconstructs the same shape, but like turned a little bit or a little bit bigger, it's fine, right? This is still the same shape. And essentially what this means is that we actually here don't want to rely on a pixel or voxel representation, because we don't actually care how this shape is kind of encoded into a grid. What we care about is the actual geometry of the object. So here we had to do things that are a little bit more fancy and not rely on classical convolutional neural networks, but also we had to think about how we train this network in a way that we don't just tell it if it's right or wrong, looking at the reconstruction accuracy. And for this, we used another types of technique that are called contrastive learning, that are also very hot in machine learning at the moment. And where very roughly, the idea is to say, okay, instead of training your model by imposing that it can reconstruct the input from the low-dimensional representation it has learned, what you're going to do is to impose a something on this low-dimensional representation. And in this context, it's to basically say, okay, I want my representation to be carved in a way that objects that are small deformation of the shape that I'm seeing stay close to wherever I have encoded this shape, and objects that are very large deformation of this shape get encoded further away. And so here we had to recycle some techniques from computer graphics. This was a lot of fun. We also discovered that a lot of the contrastive learning framework that come from classical computer vision don't deal very well with the fact that you have uncertainty in what you impose here. You know, when I'm dealing with images of cats and dogs, I can surely tell this is an image of a cat and this is an image of not a cat. When I'm dealing with cells, if I deform my cell a little bit, can I still tell that this is now a new cell type? Or can I tell that this is the same cell type? I actually don't know. I have no prior on this. So I needed to be able to do this process while having some level of acceptable uncertainty. And this is not something that was really in there. So we actually have kind of had a spin-off of this project that went into very theoretical considerations on how we adapt this contrastive learning framework to what we call soft targets, so essentially uncertainty that, again, I'm not going to talk about here, but that if you're interested, I would be delighted to tell more about. Okay, so we built this. We were excited to use this for discovery, but before we thought, let's just sanity check that what we did is not entirely insane. So what you see here is pretty much exactly the same plot I showed you for MNIST before. So what we did is to encode each and every cell in Platineris into a feature vector, then take a 2D projection here, this is UMAP, of this new representation space. And just like we did for MNIST, we color some of these points with annotations that we happen to have to see if similar things cluster together. Here, the annotations are essentially cell types annotation for a couple of cells that we got from Detlev and or other developmental biology colleagues. So again, this is not information that was incorporated in the model during training. The model doesn't have a clue about the fact it's even looking at cells, right? We're just using this as a sanity check to make sure that things we would expect to cluster together do indeed cluster together. And it turns out that they do, which is great. We can always project back each and every one of these points into the data set to see, okay, what is the actual cell that corresponds to this? We were quite reassured about that. The other thing we did to try and get another feel for the fact that the features we learn with this system actually make sense was to say, okay, we can't quite trace back features to a mathematical expression like we would for like the area or the perimeter or the curvature or these sort of things. But what we can very much do is to get a qualitative sense of what each of these features is capturing by essentially looking at data points that have high or low value for each of these features. And that's what you see on this slide for each of the three branches, the sort of low-res context feature, texture feature, the high-res no context texture feature, and the shape features. Here you have always two boxes, a blue and the red one. In the blue box, you have 4 cells that correspond to, that would have like the smallest value for the given feature that I'm showing you here. And in the red box, 4 cells that have the highest value for the feature I'm considering. So again, this is not something that gives me like an actual mathematical expression of the feature, but it gives me a sense of the range of morphologies that this feature capture. What I didn't tell you before is that we also had segmentation for nuclei separately. So we could also do this for nuclei separately, which is why you have 2 columns. But sorry, what I want to attract your attention to here is that, again, I can't quantitatively define what these features are capturing, but what I can very well do is to get a qualitative sense of the fact that this is capturing something real about morphology. And maybe the one I like the most is this one on the bottom right, because, this is essentially something that goes between like wrinkly nuclei and smooth nuclei. I don't know, yeah, that's how I would call that. And again, this is, in the end, it's a mixture of like curvature, solidity, blah, 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 lots of different things, probably in complicated nonlinear combinations. But this is actually what I think we mean when we are saying, I see morphology here and I see morphology changes, going back to my intro slide about this. So I think this is really promising because this system kind of manages to capture not one specific feature we know exactly about, but a combination of features that correspond to our visual intuition as to where morphology information is in these images. So with that, we were very confident that, okay, we can do interesting stuff with this system. And so we went back to discovery. And now what you see is, again, this UMAP of each and every cell in the Platineris, but this time it is color-coded by something different. What we did is that before the projection, so in the original feature space, we did unsupervised clustering. So essentially now what we're saying is, okay, I've encoded each and every one of these cells. I pretend I don't know anything about them. What I'm doing is just grouping things that are similar together and then looking at the clusters I obtain. And here it's something like 16 cluster or so. And now the actual biology can start, right? Because I can actually dig, I mean, not I, Detlev, and Valentina, but some people can actually dig into these different clusters and see, okay, is there interesting information there? Does this tell me something about the biology of this animal? You know, if I pretend I didn't know anything about Platinaris, can I actually uncover interesting thing with this? And it turns out that we can, because this different cluster correspond very, very much to tissues and to organs of the animal. And one thing I think I forgot to tell you about when I introduced this organism is that, okay, it has very, very stereotypical development, but another thing that it has that was very interesting for us here is that it is very strongly bilaterally symmetric, which means that if you find a cell on the left, you find it on the right as well, yeah? And this is great because it's also a very good way to validate that what we do here makes sense, huh? And I hope you can see, hopefully, yeah, that the contrast is good enough. Around the UMAP, I'm showing you some of the projections back onto some different slices of the data set of what these different clusters correspond to. And you can see that you have things like cluster 10 here that corresponds to a specific category of muscle that are indeed found on the two sides of the animal, as you would expect from the biology of the animal. But that, again, is not something we had incorporated in the system. This is essentially something that emerged from the representation, yeah? And so then there is a whole part I'm not telling you about here that Detlef and Valentina went to say, okay, we actually also have some sort of, I don't know if I have a mouse here. Yeah, interesting tiny clusters like this thing or these things. Do they correspond to cells that are functionally similar and that we didn't know about? Can we find some interesting biology about this? And it turns out that there is some interesting biology about that on like very specific types of neurons in the brain, very specific types of cells that have to do with the visual system, and so on and so forth. So the last bit I want to tell about this story is to kind of tie it back to, you know, handcrafted features. And how does that compare to what we can do with non-machine learning? And so when we were building this system, we essentially wanted to have, you know, one sort of ground truth and one sort of baseline. The baseline is obvious. This is the handcrafted features I was telling you about earlier. So everything we can measure that we kind of can define a priori and build into a big feature vector. The ground truth, well, I mean, we don't quite have one, because, yeah, nobody sat down to annotate 20,000 cells. But it turns out that for this data set, we also have gene expression that is, okay, not quite as resolved as individual cell, but good enough to give us a sense of the molecular identity of each of the cells in this data set. And so we have a kind of ground truth from like cell types as defined by cell expression, yeah? And so what you see now on the top is, again, this UMAP of each and every cell of the Platinaris encoded through our system, color-coded according to different identities as given by handcrafted features on the left, our morpho features learned from the data in the center, and gene expression on the right. And then at the bottom, you have this projected back onto, again, one slice of the data set. And the first thing I want you to see out of that is that handcrafted features are pretty damn good, honestly. And I can tell you, when we were starting to work on this, we actually struggled to beat this baseline, you know, with kind of the most basic version of what I showed you that we could think about. And what this means that, again, I think is pretty deep and interesting is that this collection of handcrafted feature, even though, yes, they are very redundant, there is a lot of information that is shared between them and so on and so forth. If you actually do appropriate dimensionality reduction afterwards to remove redundancy, we are actually capturing quite a lot of the interesting information that is present in these images, yeah, which is very promising because it means we have been doing things right from the beginning. But of course, you can get a little bit more and squeeze out a little bit more performance if you learn these features directly from the data. That's what you see in the center. You essentially manage to break down a couple of clusters into smaller subunits that gets much, much closer to the gene expression. And actually I was quite surprised, initially a bit terrified, but now I'm reassured this is actually true about the fact that we are so close, right? I mean, essentially we manage, the thing you see at the center is obtained really purely from the image, yeah? This is just essentially image-based information with no prior about the biology, and we managed to get to something that is that close to gene expression, molecular identity of the cells. So I don't think we're at a point where we can claim that, you know, from the images you can immediately find cell identity, but I mean, there is a hell lot of information contained in morphology if you image it with a modality that really shows you everything like EM does. So there is a lot of follow-up to this that is still in the making and that I'm not going to show, but we have basically from there went on in lots of different ways to try and explore further this relationship and like, you know, how much of gene expression can we actually predict from image data? How much can we essentially merge these two different representation spaces together and do imputation and so on and so forth. So hopefully in a couple of years' time, I can tell you about that. I will close with this to just use a couple more minutes and tell you the tool development side of this, because here I told you about how we develop new methods and how we enable new things that, you know, could not be done on images before. And I saw this tweet some time ago that made me reflect a lot. I let you read it while I take a drink. But that I think captures my sentiment, you know. There is one side, a very exciting and important side about bioimage analysis that is about developing new methods, being able to do things that nobody has done before, pushing the boundaries of what we can do with image data. And meanwhile, on the side, there is a lot of bioimages that are being acquired and that are being painfully processed by hand and that are being painfully processed with tools, you know, that may not be entirely appropriate because the state-of-the-art tools are very, very hard to access and to get to. And Beth talked about this much more eloquently than I could ever do yesterday. So you know exactly what I'm talking about. But I think the sort of bigger problem that is behind that is that, as we all know in this room, the academic system is set up in a way that, you know, we have a lot of resources to do new things and push the boundaries of what is possible. We have much less resources to make sure that these things are accessible, scalable, maintained beyond the lifespan of a PhD student in the lab, interoperable with each other, findable by the people who need to use them, and so on and so forth. And as I kept on producing more and more stuff with my group, this kind of became more and more a painful reality to me, that a lot of the things we are producing actually doesn't quite make it to practice because all of these aspects are not being addressed. Now, bioimage analysis is really hard. I mean, these aspects that I showed before are really hard for any field, right? But they are, I think, particularly hard to implement in the context of bioimage analysis because bioimages are so diverse, right? And this is what I showed you at the beginning with my intro slides. We have so many different imaging modalities. Like if I tell you I work with images, honestly, the only thing you know is that I'm working with a grid and that at some point there was a living system involved. You don't even know what's the dimension of this grid, right? It could be 2D, it could be 5D, and God knows what's inside, yeah? So this poses a lot of challenges around, you know, if we want to make things somewhat interoperable or standardized, we need to think hard about how we unify this data, yeah? And this is not something that the method developer are thinking about too much classically. Now, it turns out that in this bioimage analysis community, I mean, bioimaging community in general, we are extremely lucky to have had gigantic amount of efforts put towards coming to consensus on what is the information we store on these images. Yeah, how do we share these images? How do we encode what are the relevant things that we need to keep track of on how they were acquired, how they were processed, and so on and so forth. There is also a lot of work that has been put into thinking how we actually store this data in terms of like computer, right? And how do we manage them in terms of processing them? How do we come up with file formats that are as general as possible to essentially encompass the diversity of microscopy data that exists? And I'm sure many of you know about OMISAR and the push behind this. This is a thing that I'm immensely grateful for and that I find to be like one of the most beautiful success of this community, even though it's very, very early days. But like all of this is essentially background work that is absolutely critical to even start thinking about how do we do method development in a way that is interoperable and that is sustainable, right? There is a session this afternoon about this, so I hope everyone will stay there and discuss it because this is really, really critical. And the reason why I think this is so critical is because, you know, when we think about standardizing bioimage analysis, I don't think we can or even should think about standardizing the analysis process. The analysis process is something that needs to remain flexible because I think Eric mentioned this yesterday, but the rule of the game, or someone in the afternoon, I don't remember, but the rule of the game in bioimaging is that if tomorrow I want to image this thing nobody has ever imaged, I'm empowered to build my own microscope and do it, right? And I will get a new type of bioimages that I will have to analyze in a new way. And this is fine. This is what this world should be about. But what we need to do to be able to have some level of interoperability among all of this variability is to have the sort of entry point, so the data themselves, how we store them, how we share them, how we handle them, being standardized, yeah? And so because all of this fantastic effort is happening on the background, I think from the analysis side, we can start dreaming about what it would mean to build image analysis systems that rely on standardized file formats. And this is basically what motivated the University of Zurich to give funding to create this thing called the BioVision Center that I'm leading since the beginning of the year. So basically the goal of this initiative, this is in Zurich in Switzerland, is to make state-of-the-art bioimage analysis at scale accessible to all. And this really goes back to the things that Beth was saying yesterday, that if we really want methods to be actually taken on by the people who acquire the data and who are also the best place to know what a meaningful analysis is actually, we do need to make a hell lot of effort into providing framework that allow accessing these methods in a non-programmatic way and in a user-friendly way, but also that allow making these methods standard enough to operate not just on the data they were designed for, right? The other thing that is kind of part of our mission is to think about this whole notion of fairness, right? And to think about, okay, what would fair standards mean for bioimage analysis? Because there is a lot of discussion now. I mean, fair is initially mostly based on the data, right? But the analysis itself also needs to be reusable and interoperable and findable and accessible. And we very much need to think about that as a community of method developers. So we have just started very much in February, so it's very early days. But in terms of like what we are doing as a center, this is again very similar to the sort of things that Beth showed yesterday and the initiatives that she's leading at the Broad. We have a part of our activity that is about essentially R&D. So this is about developing our first project is workflow platform for bioimage analysis that is essentially based on this OMISAR standardized file format. I will talk about this more in a second. We are doing service provision, not so much in the form of analyzing data for people. What we really believe in here, and what I strongly believe in, is that I don't want to be the person analyzing the data for others. I want to empower them to analyze their data, right? So here what we do is mostly to essentially develop tools for people to be used beyond the original project it was developed for, and to support them in using existing open source solutions. And last but not least, of course, I mean, it is great to develop methods and make state-of-the-art techniques available, but only if people are also trained on how to use that properly. So this is also a part that we are doing, of course, to do training locally, mostly in Switzerland, but I mean, then also obviously other places when we have the chance to. One thing I didn't put here, because it's kind of not, it's kind of the side quest that is not officially a part of our mission, but that is equally important, is that there is a hell lot of sort of, policy making and political lobbying that must happen and that is happening behind the scene to essentially ensure that there is awareness about the need for quantitative bioimage analysis. Because in Switzerland, like everywhere else, we are at a point where everyone generates like 100 terabytes of data every day. And then, the day after is like, what should I do with it? And where do I store it? And I would very much like this to change. And this is going to be a long road, but yeah, we can make baby steps there. So because we just started, not many things are set in stone in this center, but the few things that are on this slide. The first thing, and this also echoes some of the discussion yesterday and what was said earlier in this introduction, is that we fought very, very hard to have as part of our kind of core status that all of our contributions are released under permissive free licenses. So this is BSD. This is the one that means that a company can tomorrow take my code and make software out of it and make millions. I'm fine with that. because essentially for me, it is more important that the community is able to contribute, even if these people come from industry, than to ensure that I will be the one making money. There was a lot of convincing that was needed to actually get this approved and get everyone on board behind this. It has also a lot of implication as to which kind of funding we can get. So here, I'm absolutely not saying everyone should go for permissive free licenses. What I'm saying is just that this is maybe something to keep in mind, that I think this is also a valid and important model depending on what the goals are, right? One of the really important things is that, of course, we are developing things in Switzerland for our local users, but the goal of this center is to really kind of have a way to echo what the international community is doing and to contribute to it, right? So we are very active in all the discussions about OMISAR and the specifications there. We very much want to, raise awareness locally about what's happening elsewhere in the world. And the last bit that I mentioned already in my previous slide is that the whole purpose of what we are doing is not so much to, develop yet another new method. I mean, if we do that, it's great, but it's more to ensure that the things that exist and the new things that are produced are so in a way that they get actually transferred to the users and they are actually usable to the people who do acquire microscopy images and want to analyze them. So very briefly, I just want to tell you about the kind of first big projects that we have, which is the development of a platform called Fractal. This is a platform for bioimage analysis on large data set that is entirely based on this OMISAR standardized file format. Its development is led by Joël, that you see up here, who is the head of R&D at the BioVision Center. Very briefly on one slide, the idea of Fractal is that this is a platform that is deployed on premises. So it is something where essentially a sysadmin creates a Fractal server that has access to where the data are and where the compute is. That could be the local HPC or whatever your favorite source of compute is. And then from a user perspective, you access it through a web interface in which you can build workflow, manage them, run them, add your favorite processing task like cell pose or care or whatever you like to do, any sort of quantification or restoration or segmentation or really whatever. Execute this workflow, and in the background, this essentially distributes the different tasks to be processed on the local architecture and does all of the orchestration that is needed. So from a user perspective, this is something that is meant as like not requiring an install, not requiring any knowledge on scientific computing and on software programming. And this is something that, again, is not meant that there is one server running somewhere that people access from all over the world. It is meant, okay, I just have a couple of more slides, thank you. It is meant to be something that, is deployed wherever it's needed. It is meant to process large amounts of image data in the OMISAR format. So one of the work that we are doing a lot at the moment is to actually contribute to tools to do OMISAR conversion, because as some of you who use this format may know, this is not something that is widely available yet. We are also doing a lot of work in providing tools to do easy read and write on the OMISAR format, because this makes life easier for method developer who may want to write their method as part of this platform. And the whole idea is that on the platform, there are what we call task packages, which are essentially collections of processing units to do all the different sort of types of image processing one may want to do, from which users can pick and assemble workflows as they wish. The good thing is that we are not the only one developing tasks, because that would be very unsustainable, and this is really not the goal. We are putting a lot of effort into creating this ecosystem in a way that it is straightforward. It's not yet straightforward, but it will be soon. It is relatively straightforward for method developer to put their methods into this framework, because then, okay, it's in the OMISAR format, so it's also interoperable with other framework that may do workflow orchestration in a different way, but it also hopefully relieves a little bit this hurdle of like transferring something that just got published into something that can be used, right? So what you see here are just essentially the different task packages that are available at the moment. There are a couple that we are developing ourselves, but actually quite a lot of them are not being developed by us, by other people who are not part of the BioVision Center. So this is good news. And I kind of really hope to see this grow as we move forward. Now, we do have 3 servers running at the moment, one at the University of Zurich, one at the Friedrich Michel Institute in Basel, another one at the Children's Hospital in Zurich. We have efforts to deploy one at the Sanger Institute in the UK. And there, in these different servers, the users that we have at the moment are doing things like registration, stitching, 3D segmentation here, that's organoids, or like quantification on very, very large high content imaging data. So there are quite a lot of different workflows that are working already. We started developing this. We essentially took on this project to expand it from a smaller scale project that was initially developed by Lucas Spelkman and Prisca Liberali. So this platform was very, very much designed initially to deal with high content imaging data, meaning that now it can do a lot of things on high content imaging data. If you're trying to do time series, you can't because we don't have this yet, right? So we are ramping up the capabilities of the platform, developing new tasks and also changing the logic behind to be more general. It's all work in progress, but because we really want this, we see the use of this mostly for cases in which images are so big, you can't really just, process them in a way that you actually look at them all the time. We are focusing on data modalities that generate large image volumes. So this is light sheet, electron microscopy. We're also looking at spatial expression data now. Of course, in the long run, I would want that this is available for anything from a couple of megabytes, to a couple of terabytes. In the kind of short run, we are really focusing on cases in which your image is just too big to really be opened in ImageJ, right? Now, this was a couple of slides, and there is a lot more to say about Fractal, but the most important thing to say is that this is work in progress. This is not a finished product, right? And we're not yet at a point that I can tell you, jump and use this, because this is just not going to happen. We made the choice to develop this in the open, specifically because we want to be able to incorporate feedback. We want to be able to interact with the community, and we want to be able to develop this together with the other workflow platforms that are being developed in a way that things are interoperable. So everything is on GitHub. Again, it's all under BSD3. We have documentation for the different components of Fractal. If you're interested in that, please have a look. Come to us to tell us this is crap, or this is great, or you want to contribute, or you hate it. Any feedback, really. We are purposely doing this transparently because we want to be able to exchange on that. If you're interested not only about Fractal, but about workflows in bioimage analysis with this standardized OMISAR format, we have a hackathon coming up in Zurich. This is the second iteration of this. The first one was last year. It was really fun. This one is also going to be really fun. Registration is still open until the end of the week, so please sign up for that if you want to join. We had lots of US people last year. It was great. So I would love that this happens again. And with that, I'll finish right on time, hopefully. Thank you all for your attention. I want to thank the people in my lab in Cambridge who are doing all of the hard work I showed you, and the people in the BioVision Center who are also working extremely hard. None of what I showed here would have been possible without any of these people. So yeah, thanks to them, thanks to you, and I'll be happy to take questions.
Hello, I will kick it off with an online question from Jose Moreno at the Albert Einstein College of Medicine. What would be the biggest challenges of repurposing your trained data set when changing either acquisition equipment or biological sample?
Is that a question for the first part, I guess, for more features, right? Yeah, so I think, okay, yeah, this is a great question. So the training set per se, the data set we use to train this morpho feature pipeline is this Platinaris data set, which is publicly available, right? So, this thing, you could just take it and rerun it if you wanted to. I think if the question is something like, how would you repurpose this pipeline for another data set that may have different properties? This is something that we've been exploring, because typically, I would love to be able to run this pipeline on light sheet images, but then there is no texture and it's essentially all about shape. And then I need to make sure that my system is flexible enough to essentially shut down some of the branches and not others. This is not there yet. Again, this is part of this business of making the method more than just what it is in the paper. We are working on this, and I would love this to be a more general purpose tool. It is not at the moment, but it will be one day.
Great talk. Thank you so much. First out, shout out to Fractal and OMISAR. That's amazing work that you guys are doing. I love it. Everyone should be working on it. It's great. My question is actually about the first talk. So yesterday, Eric kind of mentioned that maybe we need a foundation model for segmentation.
Yeah.
Do you think that it is even possible to construct a foundation model for morphology?
Yeah, that's a great question. I think there is a talk later this afternoon about this, so I will not steal Juan's thunder. But I think it's yeah, I think it is. I mean, it is a really, really interesting thing to think about. I'm personally not sure what a foundation model of like morphology in general would mean. Again, because this is such an undefined concept, right? And this could be 3D, this could be 2D, this could be 5D, this could be whatever. So, I mean, again, it's great to think about this. I'm personally skeptical about what a model that encloses as much variability as there is in all of microscopy would be able to learn. Now, foundation models of morphology for like specific subtypes of modalities or subtypes of problem are, I think, entirely appropriate and a really, really exciting avenue. And yeah, I think we'll hear more about that this afternoon.
Hi, this is Caterina Strambio at UMass Medical School. And thank you for the amazing talk, as usual. So talking about your self-learning models, you know, what I saw when, you know, there was this digit being compressed down to basically a feature vector. And then, you know, you can test whether or not that is good enough. I was thinking, could this also be used for like basically compressing the actual data so that we can store just the model and the low dimensional latent representation? And then if needed, we can go back to the original image. I mean, I know that this is not why it was developed, but could this, I mean, is this something that we could dream of in the future?
That's a really, really good question. Yeah, this is a really good question. I think what's a bit, so I think, again, this is really something that is worth thinking about. I think what's a little bit risky in the way that these models are built originally is that, you know, the whole reconstruction part, when we train this sort of model, is kind of really just a proxy to get a feel of like how good the representation is, because in the end, we never really look at the reconstruction part to train the model, right? And I think if this was to be used in the context of compression, then it would be much more important to actually ensure that, you know, the reconstruction is good, because this is really what we want, yeah? And I can tell you, when we train these things, I mean, okay, there are lots of different models here, and the simplest ones are not the best, but when we train these things, the reconstructions we get are, you know, they are rarely at a point that I would be comfortable doing any sort of further quantification on whatever this thing has reconstructed, right? But they're sufficiently good to make me comfortable that, okay, the information that I cared about has been encoded. And so I think that there could be some amount of repurposing of these ideas to go in the direction of what you said. And I wouldn't be surprised if some people are doing that already. I'm not aware of it. But like, I don't think that out of the box, the sort of philosophy behind this system is the right one, because it really doesn't put enough emphasis on quality of reconstruction in terms of like conserving high frequency features, for instance, which would be critical when we do compression. Yeah.
Hi, Alex Corbett, University of Exeter in the UK,
Sorry.
over here. Yeah. Thank you very much for the talk. It was great. I just had one thought, which was, what about four-dimensional data sets, so 3D plus time?
Yeah.
Could you use that if you were capturing functional data? Could you use that rather than making your life more difficult as a way of discriminating between cells that might be morphologically similar, but functionally very different?
It's a really, really good question. We have not given too much thought about 4D data, you know, in this context, because it happened that this wasn't 4D. I think one of the things that is really, I mean, I will say something and you tell me if that answers your question or if this is too tangential, but like one of the things that I've been really interested in the context of other projects with 4D data is that, you know, the time component, again, for lots of reasons, we treat this as a very separate dimension from spatial dimensions. But again, from a computational and conceptual point of view, it is essentially just one more dimension, right? And so I think there is a kind of broader, interesting thought to have here about how would you encode 4D data not as like 3D plus time, but as really like 4D manifolds that you encode with this sort of systems, right? And I mean, shape in 4D is, for me at least, easier to think about because again, okay, we can't quite represent it, but this is essentially, yeah, it's essentially a manifold, right? So it's something I can define geometrically. Texture in 4D, I'm not exactly sure how I would define that, but I think thinking about representations of texture in 4D, for instance, is the right way to go if you want to then run through this sort of encoding approaches to treat these data sets as genuine 4D and not like 3D plus an extra sort of series on top. Yeah, I don't know if that kind of answered or if I went too far, but
No, that's
yeah.
great. Thank you very much.
Thank you.
Do we have time for one more? All right, one more. Here you go.
Eric Waite from Elephas here in Madison. How do you think about error propagation? So if we're training models that are built on models that are built on models, so like if you use some model to run your segmentation, then you feed that into another model. If we've used some more handcrafted, you can kind of throw out or try to manipulate the data in a way that you know it's good quality going in.
Yeah.
How do you think about that?
Yeah, no, absolutely. I mean, so I've never been doing a lot of segmentation in my life. So I think, I mean, I know this is something that in the segmentation world, people think a lot about. In the quantification world, I think this is really important to think about as we start going to the direction that was mentioned earlier of like, you know, if we had things like a foundation model of morphology that we kind of just reuse over and over again, I mean, how do error propagate there? And this, I mean, I don't have the answer to this. I think this is, again, what's really interesting there is that in the context of segmentation, we have very good metrics to quantify errors, right? Because we have been thinking about this for a long time, and we have been thinking about how, you know, dice is not so good and intersection over union is not so good and this thing is better than that other and whatever. I don't think we have been thinking too much about this in the context of quantification because what would, you know, a good quantification, a correct quantification mean is like completely problem dependent really and completely dependent on like the question you're asking and the data set you're looking at. So, I mean, I guess the short answer is that I don't even really know how to think about that properly, but I think this is something we need to start thinking about as we move to this data-driven model and as we move towards reusing model for quantification specifically. I think for quantification, for segmentation, sorry, it's, again, I wouldn't say this is sorted, but I think we have frameworks to be able to assess how you know, valid is a reuse of a model or not. I don't think we have any of that for quantification yet. No, thank you.